Hello friends. So here we are going to discuss extracellular matrix and associated disorders. So please like, share and subscribe to my channel. So what is extracellular matrix? It is a non-cellular component of all tissue and organs. So it is a non-cellular component and it is present in all tissues and organs and enumerate various component of this extracellular matrix. So here you can see the most important component is water, then fibrous proteins like collagen and elastin, glycoproteins like fibrillin, fibronectin and laminin, and proteoglycans like glycosaminoglycans. So these are the important component of extracellular matrix. So here we are going to discuss the functions of extracellular matrix. So extracellular matrix that holds cells together and provide a porous pathway for diffusion of nutrients and oxygen to individual cells. So here you can see this is your extracellular matrix where you can find you can see this composition here which contains fibronectin, proteoglycan gel, collagen, and here this extracellular matrix it holds cells and it also provide a diffusion pathway. So here, this extracellular matrix, here you can see there are some minute small pores are there, and that pores are responsible for diffusion of nutrients and oxygen to individual cells. These are the cells, and here from this blood vessel there is a transfer of these nutrients and oxygen to individual cells. Next function is extracellular matrix protect oxygen and it provides protect organ and provide elasticity to organ. We'll see in the next slide. It also regulates growth, migration and differentiation. The cellular growth, this migration of cell and differentiation, it is regulated or it is maintained by this extracellular matrix. And the protein and glycans present in extracellular matrix, it forms a gel-like structure and it functions as a cushion and lubricate against a mechanical shock. As in case of joint, we will discuss in next slide. So, so here you can see the heart, and here this is a the tissue which contains extracellular matrix, so it protect your organ also, and also the elasticity of the organ it is provided by extracellular matrix. Then it also participate in a cell growth, cell proliferation, differentiation, and migration. It is done by extracellular matrix. Here you can see there is an increase in cell size. It is called as a cell growth, proliferation, and differentiation into the different types. Then, as we already discussed, that protein and glycosaminoglycan present in extracellular matrix, it act like a gel-like structure and it act as a cushion and lubricate against a mechanical shock. So these are the articular cartilage which contains, which is made up of our extracellular matrix, and it protect joint and it is bone against friction, and this articular cartilage which reduces friction here. So these are the functions of extracellular matrix, and it helps against injury. So the in extracellular matrix, the first example is the collagen. So in this lecture, we are going to discuss this collagen synthesis and associated disorder. So here, this structure is nothing but a collagen. Okay, tail. So the cell which is required for the collagen formation is a fibroblast. And in the nucleus of this fibroblast, we have a DNA which contains a gene for collagen. So by the process of transcription and translation, there is a formation of alpha chain. And this alpha chain which contains glycine, X and Y. These are the repeated sequence you will find in a collagen. So X it is always a proline and Y it is maybe proline or lysine. So here in this alpha chain, the maximum amino acid you will find that is a glycine and proline, and why it may be proline or it may be a lysine. So this alpha chain, it undergoes hydroxylation reaction. For that, the coenzyme is a vitamin C, and the enzyme name is a prolyl 4-hydroxylase and lysine hydroxylase, which are responsible for hydroxylation reaction. So after hydroxylation, before exit of this alpha chain, it undergoes glycosylation. Again, for glycosylation, we require coenzyme vitamin C. So here there is an enzyme is glucose transferase or galactose transferase. So transfer of this glucose and galactose to hydroxylysine of this uh, alpha chain. So these are the two prerequisite steps, hydroxylation and glycosylation, which are important for the alpha chain formation in a collagen synthesis. And this step, it takes place in a rough endoplasmic reticulum. Before exit, here this there is assembly of this pro alpha chain takes place in a Golgi apparatus, and after assembly of this three alpha chain, this three pro alpha chain into the pro collagen. This assembly takes place in a Golgi apparatus, and after that it undergoes exocytosis. Means what? There's a expel or this pro collagen, it comes out of cell of this uh, Golgi apparatus, and here you can see we have a two terminal carboxyl terminal and amino terminal. So here the enzymes like peptidase like carboxypeptidase and amino peptidase participate here, and after that you will get the collagen. Let's see how does it happens. So here we have a procollagen N terminal, procollagen C terminal, and the procollagen peptidase, which are responsible for giving you a proper collagen, that is a type 1 collagen. Now, now here you can see that the collagen, it is a release out, and this collagen, which is a monomer, which is also called as a tropocollagen, it has to undergo assembly. It is called as a spontaneous assembly through a covalent cross-linking so as to form a mature collagen fibers. Such collagen fiber confirms strength and rigidity on the collagen fibers. And this covalent cross-linking, it is done by the additional interchain hydrogen bonding where hydroxylysine and lysine residues are responsible for forming this cross linkage. So the enzyme is a lysyl oxidase, which is a copper containing enzyme which is responsible for forming a covalent cross linking. So here you will find a covalent cross linking, and here your important enzyme is a lysyl oxidase, which is a copper containing enzyme, and ultimately you will get a collagen. So collagen, which is the most abundant connective tissue made up of three polypeptide alpha chain, and each alpha chain contains 1000 amino acid. Each chain is called as the alpha chain, and the three main amino acid, that is the glycine, proline, and Y, that is the hydroxyproline or hydroxylysine. So these are the important steps in a collagen formation. So what are types of collagen? Type 1, which is present in the bone, skin, and tendons. Type 2, which is present in the cartilage, vitreous body, and nucleus pulposus. Type 3, which is present in the fetal skin and connective tissues. And type 4, which is important, which is present in the basal membrane or basement membrane. So type 1 and type 2, these are the important collagen, which as type 1, which is present in your bone, skin, and tendons. Here, this the maximum part of this collagen or this compound, this fibrous protein, which is mainly seen in the skin, bone, cartilage, tendon, blood vessels, cornea, teeth, heart wall, and basement membrane. Out of that, the maximum percent is seen in the skin. It is the 74% of collagen which is present in our skin, 64% present in a cornea, and 50% present in the cartilage, followed by 23% present in our bone, followed by liver. So these are the important functions of collagen which participate from maintaining the structure of particular tissue, and it also helps in preventing the friction, and it is an important part in a basement membrane or basal membrane. So what are the associated disorders of this collagen? So first one is the scurvy, second one is the osteogenesis imperfecta, and third one is the Ehlers-Danlos syndrome, and fourth one is the Alport syndrome. So we'll discuss it one by one. The first one is the scurvy. So scurvy, as we know that whenever there is a deficiency of vitamin C deficiency, when it will happen? Whenever your fruits intake is less. So as we know that vitamin C, which is required for the hydroxylation reaction, as we have seen in this step, the hydroxylation reaction we require vitamin C. If this step is not happening properly, so that there is a decrease in a collagen synthesis. The collagen synthesis will not occur properly, and because of that, the manifestation of scurvy will be there. This is the biochemical basis of scurvy because of deficient hydroxylation reaction. These are the manifestation, that is the bleeding of gums. You can easily get injured, and there is a pain in your teeth whenever you tap on your teeth. Then second one is the osteogenesis imperfecta. Here there is a defect in a protein called as a type 1 collagen. Why there is a defect in protein? Because here there is some defect, because there is defect in assembly. The assembly is not happening properly, as we know that there is the pro alpha chain that undergo assembly so as to form a pro collagen. If this assembly is not happening properly, it means that your collagen will be defective. So here you can see that normally there is a glycine which is present in a pro alpha chain, but in case of osteogenesis imperfecta, the glycine is replaced by cysteine, preventing the correct folding of collagen fibril into the triple helix. So here there will be no proper folding because glycine is replaced by cysteine. Here you can see this is a normal collagen, but because of there is a no proper folding, this collagen, this pro alpha chain will be like this. So it is seen in a osteogenesis imperfecta. So as we know that the collagen gene, this is a collagen 1A1 or collagen 1A2, these are the two genes which are responsible for type 1 collagen synthesis. If there is a defect in this collagen formation, your bone become brittle, easily breakable. As we know that collagen which is present in eye, that is the sclera. So here your sclera will appear blue. And the manifestation, second, third one is the deafness. Deafness is there because there is a your ossicles is also made up of bone, and there is an improper development of ossicle responsible for deafness, responsible for hearing loss, and here your teeth become opalescent because of the proper collagen which is required for development of teeth is not there. So here you will find blue sclera. The multiple fractures are there because of the brittle bone. Your teeth is not proper and hearing loss is there. So here you can manage it by the bisphosphonates. Third one is the Ehlers-Danlos syndrome, and it is a type 3 collagen deficiency. And here the deficiency of pro-collagen peptidase and lysyl oxidase. What is the biochemical basis of this Ehlers-Danlos syndrome is the peptidase enzyme which is responsible for cleaving N-terminal and C-terminal end of pro-collagen. So this enzyme is deficient so that the proper cleaving will not there. And second thing is the deficiency of lysyl oxidase. As we know that this lysyl oxidase which is also responsible for assembly, this assembly and the formation of covalent interlinking, the covalent bond which is formed between the lysine, that is hydroxylysine and lysine of this collagen, that will be defective, which is responsible for Ehlers-Danlos syndrome. So it is not there. Initially it was there, but there is no such covalent cross-linking seen in a Ehlers-Danlos syndrome. So such patients, there is a hyperflexibility of your skin. It is commonly seen in a classical type, and there is hypermobility of joint. It is the seen in a classic type of Ehlers-Danlos syndrome. In muscular type, your vessels are there is a formation of aneurysm, means there is a dilatation of your blood vessel. It is seen in a patient who are suffering from vascular type of Ehlers-Danlos syndrome. And the last one is the Alport syndrome. Here there is a type 3 collagen which is involved, and patient present with a hereditary nephritis, and it is a X-linked disorder. So here there is a mutation of gene collagen 4A3, 4A4 and 4A5. Three of these six human genes are involved in a basement membrane, and because of this mutation there is the improper development of basement membrane. So what are the features of this Alport syndrome? It is hematuria, sensory neurodeafness, and conical deformation of anterior surface of lens called as a lenticonus. So these are the pathognomic. In exam, the question may be asked: what are the features of Alport syndrome, and what is the defect? So the mutation in a gene which are responsible for collagen formation, that is the 4A3, col 4A4, and col 4A5. And the collagen involved is a type 4 collagen, which is responsible for formation of basement membrane. And these are the clinical features. In case of Alport syndrome, the basement membrane of kidney, which is present in the glomerulus, that is not properly formed because of defect in a type 4 collagen, because of the mutation in the alpha chain, the gene which is responsible for formation of this alpha chain. So here there is a nephritis is there, and the RBC that can easily appear in urine. That's why there is a hematuria seen in a Alport syndrome. Then next condition is there is an organ of aorta where the basement membrane is not developed proper. It is also responsible for sensory neural deafness because of the defect in a basement membrane of this organ of your ear. And this is a normal lens which is present in our eye because there is a defect in the lenticular basement membrane. It is because of the Alport syndrome where your lens it become conus like. That's why it is called as a lenticonus. The anterior surface of lens it is like a cone. That's why it is called as a lenticonus. So here in this part we have discussed three disorder. One is the scurvy, second one is the Ehlers-Danlos syndrome, Osteogenesis imperfecta, and Alport syndrome. So in exam, the question will be asked which type of disorder, which because of defect of which collagen responsible for which type of disorder. Enumerate any four disorder or two disorder of collagen uh, defect in because of defect in the collagen, and the question another question will be the clinical feature. So this is all about collagen and its associated disorder. In next part, we will discuss elastin and associated disorder. So please like, share, and subscribe to my channel. Thank you.